Hello everyone, and welcome to today's live broadcast, Optimization of UCB's Transient Expression Platforms, presented by Christina Gordon, Scientist of Protein Sciences at UCB Pharma. I'm Susie Valdez, and I will be your moderator for this educational webcast, presented by LabRoots and sponsored by Gibco, part of Thermo Fisher Scientific. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during this presentation. Just click on the green Q&A button located at the lower left of the presentation window and type your questions into the box that appear on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing this presentation in a slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have any trouble seeing or hearing this presentation, please click on the support button at the top right or use the Q&A button and let us know that you're having a problem. Please join me now in welcoming Christina Gordon. I will now turn the presentation over to her. Welcome, Christina.
Yeah, hello everyone, and Sue, thank you very much for the kind introduction. In the next 20 to 30 minutes, I'm going to talk about the optimization of UCB's transient expression platforms. So I'm going to introduce you to UCB, to our transient expression platforms at UCB, to the improvements that we made to our large-scale transient expression platforms, the improvements that we made to our small-scale transient expression platform, and our plans for automation. So UCB is a global biopharma company, and our vision is to meet our patients' unmet needs. And we're focused on severe diseases in two therapeutic areas, namely neurology and immunology. We have over 1,000 employees within UCB R&D, spread over 2 research centers based in Slough. And if you look at the top left corner and the middle one, those are our sites in Slough. And then in Bren in Belgium, which you can see at the bottom right corner. UCB spends 27% of its revenues within R&D, which really shows how research-driven we are. So here I wanted to show you our full and exciting UCB pipeline. So throughout the project progression, you can see that we've got quite a number of exploratory targets, about 10 to 20 in the early stages. Then we have our early stage projects, about 15 to 25 at any one time, through to the preclinical phase. And then in our clinical phases, we've got quite a number of neurology projects, here seen in red, and immunology projects, here seen in blue. And what I wanted to show you here is that we have quite a number of projects, and they all will need to be supported by a large number of reagents during their progression. And this is why I wanted to also show you a typical project progression and the reagents that are required to support the drug discovery process. So a typical progression would be from lead identification through to the clinical development. And during that time, there are large numbers of reagents required. As you can see here in the early stages, during the lead identification, large panels of antibodies and antigens are required for small amounts. And then going through to the clinical development, the number of antibodies required decreases, but we need larger amounts of those to support all the various studies at any one stage. And we typically would provide these reagents through transient protein production, which takes us about 2 to 8 weeks, through to the preclinical phase, from the DNA to the purified protein, and from then onwards, with a slight overlap, it's the stable cell line generation that kicks in, which takes about 3 to 5 months from the DNA to the final purified protein. And all of this really shows that there is a clear need for flexible and efficient transient expression platforms to fully support our pipeline. So here I just wanted to show you a range of antibody formats that tend to be required, and the choice of format is usually driven by the therapeutic need. And in the past, we've mainly been asked to express your standard antibodies like your IgG, which tend to be easier to express. But more recently, we've been asked to express bispecific formats and other antibody formats, which tend to be more difficult to express. So this really shows that our expression platforms need to be really capable to support all the needs. So here I'm going to show you the expression platforms that we have available for the medium to larger scale, and one of them is our in-house electroporator. You can see a picture here on the side. So a typical process would be that you take your cells, wash them in buffer, concentrate them at your DNA, you aliquot them out into the cuvettes, and you can see one of these cuvettes in the cuvette holder in the picture, and you electroporate your cells, and then you transfer it into your production media. This is a fairly efficient method that we have, and we're typically using this between 200 milliliter up to the 5 liter scale. It takes 4 milligram DNA per liter transfection. About 14-day process, and we're seeing quite high yields of around 200 to 1,000 milligrams per liter antibody. The slight downside of this method is that it becomes really labor intensive if you do transfections above the 5 liter scale, because a 10 liter expression, for example, would take 120 cuvettes. For this reason, we brought in the Maxite large-scale flow electroporator, which you can see in the picture here. So for a 10 liter expression, this electroporator would take about half an hour. So the way this works is that you again take your cells, you wash them in buffer, concentrate them at your DNA, and they are collected in that bag that you can see hanging on the side of the machine. The electroporator would then pump the cells through the electroporation chamber, which you can see in the middle, slightly orange, and it will collect the electroporated cells on the side, waiting to be transferred into your production medium for culturing. And this method we typically use above the 5 liter scale, and we again need 4 milligrams of DNA per liter transfection. It takes the same amount of time, 10 to 14 days, and due to intensive optimization that has gone into this system, we are also able to achieve around 200 to 1,000 milligrams per liter antibody. So now I wanted to show you some of the historic improvements that we made to our large-scale platform. We've been continuously optimizing this platform over the years, and about 10 to 15 years ago, we started to develop the platform, and we found that a CHO suspension host with electroporation and a temp shift applied, adding expression enhancers would give us the highest yields. Throughout the years, we started to do host cell line engineering, and we developed our CHO SXC cell line, which overexpresses XBP1, which results in enlarged ER, and ARO1 alpha, which aids disulfide bond formation. And thereby, we are most of the time seeing up to a 5-fold increase in expression levels. And moving on through the years, we also look continuously at optimizing our media and processes. And here is one example where we looked at different commercially available media. And in our case, ProCHO5 has given us the highest yields. And another example here is that we developed a fed batch process for our CHO electroporation process, and this fed batch process has doubled basically our yields over our standard process. So some of the more recent improvements that we made to our large-scale platform is that we revisited the media, additives, and feeds again, and we found a new production media that would give us about 1.5-fold increase over our current media. And previously we have been using sodium butyrate as an expression enhancer, but we found a recent paper that states that valeric acid can improve recombinant antibody production. And we tested this, and we found that when we're using valeric acid in our system, we can see up to a 1-fold increase in expression levels, which is great. And then finally, we also optimized our feeding strategy, and by doing so, we increased our yields. Sorry about that, that's our fire alarm. Okay, it was just a test, sorry about that. So by doing so, we've increased our yields by about half-fold. So when we look at the impact that these improvements had on our large-scale expression system, you can see this graph here that shows the amount of antibody expressed over the past 4 years. And if you look at 2013, where we expressed about 40 grams, we are in 2016 drastically up at about 140 grams, which is a drastic improvement over the years. So now moving on to our small-scale systems. So throughout the years, we've seen a large increase in the number of small-scale transfections, and we believe that even more will be required in the future. So this graph is actually an example where you can see that in 2013, the transfections performed that were below 30 mils are quite low. And then from 2014 onwards, there is a large increase in the numbers of small-scale transfections below 30 mil. So therefore, we felt that we need to increase the throughput of our small-scale expression system, and there is definitely scope for improvement. So our aim was to develop a high-throughput small-scale expression platform, and for that, we wanted to have a look at the host again, find an optimal one, see which transfection method is the most suitable one, find a suitable culture vessel, and also try to improve our clarification method. So now I just wanted to quickly tell you which transient expression platforms we are using currently for our small-scale systems. So one of them is the reagent-based system called XP293, supplied by Thermo Fisher. I just wanted to quickly show you what the process is. So you take your cells, dilute them in XP293 medium, you take your DNA and your reagent and dilute it in Optimum, you add them together, incubate it for 20 minutes before you're adding it to your transfection culture, and after about 16 to 18 hours, you would add your enhancers. And this is quite an efficient system, which we mainly use at small scale at around 500 mil, up to like about 200 mil scale mainly, but we have been able to push it up to the 10-liter scale quite successfully as well. It takes less DNA, only 1 milligram DNA per liter transfection. It is also a shorter process of about 7 to 10 days, but it gives us slightly lower yields in comparison to our other systems, only 10 to 600 milligrams per liter antibody. We also have another system, a reagent-based system that we use mainly at small scale, also supplied by Thermo Fisher. It's called XP2. It's very similar. The process is very similar to the XP293 system. So here again, you dilute your cells to about 6 million cells per milliliter, and you take your DNA and your reagent and dilute it in a medium called OptiPro. You mix them together, and quite straight afterwards, you add it to your transfection culture. On day 1, you have the option of reducing the temperature to 32 degrees and to add feeds, and then there's another option to add more feeds on day 5. For this system, we mainly use at about the 300 milliliter onwards mark, have been able in a few occasions to test it up to the 10-liter scale. It takes also only 1 milligram DNA per liter transfection, process time similar to our CHO electroporation method, 10 to 14 days. But we've seen higher yields here as well. We've seen between 200 up to 1,000 milligrams per liter antibody. So if we're moving on now to finding the optimal host for our high throughput approach, we compared yields of CHOs and HEK293s at small scale. Here you can see that quite consistently, the CHO cells would give us higher yields, which is why we decided to move on with our CHO cell line. So if we're looking at the selection of the optimal transfection method, we have 2 methods available. At the moment, it's our CHO electroporation and a CHO reagent based. Both of these giving us fairly high yields. So the question was, are they also easy to automate? And we considered quite a few different options regarding the electroporation method, but we felt that for now it wasn't really suitable for an automated high throughput approach. So we wanted to see whether the reagent-based system would live up to this. So to test this, we felt that we already have the Amber 15 in-house. It's an automated liquid handling and culturing system. So we wanted to program this automation system to do the transfection mix preparation and the culturing for us. So here's an example experiment where you have your shake flask control on the side, as you see in purple, and then we have programmed the Amber to do the transfection mix preparation for you with its liquid handling capabilities, and then we took the prepared transfection mix off the Amber and added it to shake flask cultures and incubated it in the incubator. And as you can see here, the 6 identical transfections, there is scope for improvement, but they're not too far off our shake flask control. So now we did it the other way around. We prepared the transfection mix manually in the hood, as we would usually do, and then added it to the automation unit, the culturing unit of the Amber. And as you can see here, there is a clear benefit over doing so. And then finally, we actually programmed the Amber to do the whole job for us. And as you can see here, in most of the cases, if not all, the yields are good, better, if not better, than our shake flask control. So this made us feel very confident that the reagent-based method is the one to go forward with. So now looking at the selection of an optimal culture vessel, and we felt like the Amber 15 is an automated system, but it isn't high throughput enough for us. And also the 10 milliliter scale is not quite sufficient enough. So we were looking at something around 30 to 50 mil scale, and we screened the market and tried to find an optimal culture vessel, and we found that spin tubes are ideal for this high throughput approach because they require less incubator space, and they're also easier to automate because there are already devices out there on the market who can handle these type of tubes. And to test this again, we brought in spin tubes from 3 different suppliers, here supplier A, B, and C, and we compared these to our shake flask model. And as you can see, all of these spin tubes performed really well in comparison to the shake flask. And we felt like we wanted to move on with the spin tube supplier A. So finally, we wanted to improve our clarification method, and to do so, we compared the standard versus a new method developed by Sartorius. And if we look at our standard method, where we centrifuge our sample for at least 30 minutes at high speed, and then stereofilter it through a 0.2 micrometer stereofilter, we quite often find that the filters would block, and several filters would be needed to clarify the sample. So here we have one example where I took a 1 liter culture, divided it in half, and 500 ml. I centrifuged and added the supernatant to a bottle top filter, and you can see that after about 100 ml, the bottle top filter blocked and nothing was going through anymore. So if we look at the DE clarification method in comparison, there's no centrifugation needed. And for example, you would draw up your cell suspension into a syringe that is already pre-filled with the diatomaceous earth. You gently mix it, and then you push it through a 0.2 micrometer stereofilter. And this is fairly easy and most often does not result in any blockage. And this is also the reason why it would be more suitable for automation. So I've got the direct comparison here. So the other half of the culture, I took and added the DE to it, mixed it gently, and then added it to the bottle top filter straight. And as you can see here, all the supernatant went through without any problem. So I just added some pictures as well of a smaller scale approach using DE. So here you have your pre-filled syringe with DE. You draw up your cell suspension from your culture vessel. You mix the syringe slightly, and then you add your sterile filter to the top of the syringe, and you can just push it through, and you receive your clarified supernatant. So for these reasons, we felt like the DE clarification method is definitely more suitable for a high throughput automation approach. So now coming back to our initial plans for increasing the throughput of our small-scale expression system, we found that the CHO cell line would be most suitable, that the reagent-based method is suitable for this approach, and that spin tubes would be ideal. We also found an improved clarification method. So the next step would be to put all of this into automation. So our future transient automation platform, we're imagining to be a modular system where you have a transient expression module which can do your preparation of your transfection mixture, will do your DNA calculations and dilutions, it can do the addition of your cells, the feed additions, the cell count via an integrated cell counter. It would do sampling for assays, and all of this at a culture scale of between 20 and 40 milliliters. So we have a module supporting this transient module would be the DNA preparation and quantification module, which we imagine to be automated. Then this module would also need to be supported by the cell suspension preparation, which at the moment we're imagined to be manual still. Then there needs to be a user interface where you can add media and feeds and samples for another analysis. Then you would have the interactions with your incubators. That could be either manual or automated. And finally, also a clarification module, which we imagine to be a DE-based method. So just on summary, I've shown you the ever-increasing demand for proteins to support the UCB pipeline. I've shown you the continuous improvements that were made to our transient expression platforms. I've shown you our robust and high expressing CHO system for large scale and our high expressing and high throughput system for small scale. And the next steps would be for us to increase automation to increase the small scale delivery. So I would like to thank the protein science team at UCB, especially Bernie Sweeney, Sam Hayward, Hannah Hailu, and Kathy Kane. And I'm very happy to answer any questions. Thank you very much.
Thank you so much, Christina, for that informative presentation. We will now start with our live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of your presentation window, type your questions into the box that appear on the screen, and click the send button. We'll answer as many of your questions as we have time for. So let's start with our first question from our audience members. Christina, you mentioned that there has been a large increase in the number of small-scale transfections in the past few years with the prediction of further increase in the future. How are you going to ensure that the automation system can cope with this demand?
Yeah, thank you very much. That's a great question. We have decided that we wanted to keep this automation platform as simple as possible. So we wanted to just focus on the capacity rather than focus on how many different things it can do, how many different scales, and so on. So if we keep it simple and say it can do consistently 30 mils all the time, and if we ever had a demand, for example, of expressing 200 mils, then we would just do several times 30 mils. So we wanted it to be as high throughput as possible, but fairly simple. So hopefully that will work.
Thank you for that. In your talk, you also mentioned that you are using both the XP293 and the XP-CHO reagent-based systems at small scale. The XB-CHO system seems to achieve generally high yields. Under which circumstances would you still use the XB-293 system over the XB-CHO system?
Yes, thank you for that question. So the XB-293 system is still a great system because, especially when there is time pressure, it has a shorter process, shorter time. So it's only 7 to 10 days. So we would definitely use it when we're under time pressure. And the other thing is the translational modifications. So sometimes there's an advantage of using human cells over CHO cells. And just thinking of another one is, yeah, so we found that not every antibody or bispecific would express as well in CHO cells as it would do in HEK cells. So sometimes we just make the decision to use the XB-293 system instead of the CHO system for that reason, because some proteins express better in the 293s.
Wonderful, thank you for that. We do have one more question from our audience members. Can you explain one more time the sartorius DE filtration? Are you adding DE to culture supernatant and filter the mixture through bottle top filter? What is the pore size of the filter you're using?
Okay, so DE, just wanted to explain what DE is. DE is diatomaceous is actually like has a porous structure, and what happens if you mix it with your cell culture, the supernatant, it would just impact your cells within the DE structure and would still keep it porous so that you wouldn't receive or wouldn't get any filter blockage. So when you're using DE, a 0.2 micrometer filter would be sufficient with that, but you need a filter that has a little bit of dead space on top, because there will be a filter cake that is made out of DE and your cells and your debris.
Thank you so much. If there are no further questions, we want to ask Christina, do you have any closing remarks you'd like to share with our audience members today?
Yeah, thank you very much for this opportunity. I really enjoyed it, and I'm very thankful that I got the opportunity to present here in this forum.
Well, thank you very much, Christina, and thank you to the audience. I'd like to also thank LabRoots and Gibco, part of the Thermo Fisher Scientific, for making today's educational webcast possible. Additional questions will be answered and published on Thermo Fisher Scientific's website. You will receive an email notification when answers are available. Before we go, I want everyone to know that today's webcast is available on demand viewing until August 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live events. We have about 25 minutes to take a break, stretch your feet, till our next fabulous presenter begins. Thanks so much for joining us at this virtual conference. We hope to see you again. Goodbye.